When we are talking about gut microbiota and its relationship with the GLP-1, that how this gut microbiome works through GLP-1, let's see some basic actions of GLP-1 or incretins. We call it, there is a GIP, PYY, there are so many gut hormones or gut peptides which are secreted in response to food, and then these incretins stimulate beta cells so that there is a more insulin comes, and this response happens only when the glucose is given orally. It doesn't happen when you give intravenous glucose. So that is called as an incretin effect, that more insulin comes from the beta cells than expected compared to IV glucose. So this incretin, then now everyone knows, is actually diminished effect in type 2 diabetes, and the same kind of GLP-1 which we need, and you can see here that effect of GLP-1 is glucose dependent, and the glucose goes down because of the GLP. There is more insulin comes from the beta cell, and the same time incretins act on the alpha cell, so there is a glucagon suppression which happens simultaneously. So this lack of glucagon suppression, again it is very important in type 2 diabetes, which contributes to hyperglycemia and more hepatic glucose output, as we all are aware of. So a diminished incretin response in type 2 diabetes, or maybe because of obesity, and we will come to that in our next discussion, that how to improve this GLP-1 response. It's very interesting that this GLP-1 as a treatment, which actually Drucker has he started his research, was found from the saliva of Gila monster, which is again it's a Gila monster, which is a big lizard, and it can remain without food for many months in the burrows. So comes out and eats occasionally and remains without food. That is because of very high GLP-1, which actually controls the food intake. And if the GLP-1 is high, you eat less food, and that was the beginning of his research. And then of course we all know Exendin 4 molecule was researched, and now we have so many good GLP-1 analogues and GLP-1 based therapies for treatment of type 2 diabetes. What it does is that when the food enters the digestive system, the incretins are secreted from the L cells of the intestines. They have an effect on pancreas, and they secrete insulin from the beta cells, again which increase glucose uptake in the adipose tissue and the muscles, as well as it improves the glucagon secretion is suppressed. All these contributes to decrease in the glucose, and that's how it helps. Whenever there is an incretin effect, the glucose goes down, there is an increased gastric emptying, and it also acts on the brain, which improves satiety, and the food intake goes down, and that's how the incretins work in the normal physiology. Coming back, how these incretins, so we know we know lot of good effects of incretins now, that they can have a weight loss effect along with good glucose control, they reduce fat, and now there are good cardiovascular outcome trials which are showing that it protects heart, it decreases atherosclerosis, and that is the importance of GLP-1 analog or GLP-1 based kind of a treatment. But the endogenous GLP-1 acts through this, and it's very important for a normal metabolism and for the body. So how this GLP-1 is related to gut microbiota, and there are mechanisms linking gut microbiota and GLP-1 secretion. So you can see from these slides that when there is a dietary fibers which are not soluble, and we will come to that when we are discussing this with Ruchu, that these dietary fibers get fermented in the big colon and they produce short chain fatty acids. And in the lining of the gut, there are some L cells here, and these short chain fatty acids actually act on the L cells and produce GLP-1 secretion or improve that. You can see in this slide, which looks better, that gut microbiota, when it is healthy, it acts on the L cells by producing short chain fatty acids as well as other nutrients. So this is a enteroendocrine cell which is dispersed. This also number goes down with obesity and diabetes, and they have receptors here in this border, and the receptors work in response to different nutrient, lipids, carbohydrates and proteins, and short chain fatty acids, they get attached to the receptors of L cell and secrete GLP-1, which goes into the blood and has all its good effects to decrease food intake as well as weight loss and improvement in the glucose. Also, if you know, through carbohydrates, this GLP-1 is secreted through SGLT1 receptors. That is also, there are other kind of a stimulus from the gut, from different nutrients through gut microbiota, which can stimulate these L cells and increase GLP-1, and that's how this is important. So, whenever there is a disruption in this gut microbiota, I think there is a problem. So, I would like to share a 2020 research paper which was just published from China, and it says that implications of the gut microbiome composition of type 2 diabetes patients from northern China. What it says, it shows that they analyzed the gut microbiota from a cohort of healthy and diabetic individuals from the northern China, and what it showed that a significant decrease in the gut microbiota diversity of diabetic patients as compared to healthy individuals. And so that was a very important first kind of a large cohort based or a population based study, which is actually showing, the small studies were there, but this was the first of its kind, which is actually showing how the diversity is changed. So, I want to ask Ruchu that these large number of microorganisms, there is some different types, and I think this proportion is very important for this. So, can you elaborate on this?
Yeah, that's very true. Basically, in our body, there are different type of microbiomes.
So